the introduction, Sharon. So my talk today is going to talk about how we're using some microprobe spectroscopy techniques and to look at sulfur, and in particular some of the analytical challenges for interpreting that spectroscopy. And that's hopefully maybe able to inspire some future thoughts of how DFT type calculations could be used to further explain the science and topics that we're looking at. In this sense here, we're trying to describe a distribution and origin of different phases across geological length scales, so the entire sedimentary basin, to try to understand that chemistry, but then looking at the relationships of chemistry and structural bits at the micron scale to try to understand when things were deposited and what's happened throughout geologic time afterwards. And so this is our multi-scale challenge of trying to understand the micron scale and extrapolate that up to the basin and geological scale. So we're doing this at Sulfur. So this is our Beamline at SSRL. This is Beamline 14.3. It's the new and revamped 14.3. So there's our brand new sample chambers and new axially symmetric focusing optics that are increasing fluxes and reducing our spot size. So if you haven't seen 14.3 and haven't seen it lately, this is the new look and feel. And the approach we're going to use here is kind of a combination of both imaging and spectroscopic techniques. So typically, when we talk about micro XAS and XAS imaging, we're looking at larger areas, and I use that in quotes because large might be just a square millimeter or two, but looking at composition maps that you've seen previously and some elemental correlations. And then we go on to small discrete points and look at things like XANES or XFs. What we'd like to be able to do often here is combine these two techniques together and create an image map of oxidation state and or species. And sulfur is a fantastic way, a fantastic element to do this on. I usually admit that now that sulfur is my second favorite element behind manganese, which was my first elemental love. But sulfur has a great possibility for doing these types of measurements because you have a wide variety of oxidation states over a wide energy range. And so we can do this technique of selected excitation energy XAS, chemical imaging. There's about 100 different names for this types of idea or XAS imaging. But we take maps at several energies, excitation energies, across the absorption edge and examine the fluorescence yield and do these at typical energies that could be used to differentiate the different species. And again, sulfur has this very rich this distribution of pre-edge features and post-edge features that really makes looking at redox states in sulfur compounds very interesting. Also makes it very complicated. And this is, I think, one of the reasons why computational methods could really come into play to help describe some of these chemical species that we're seeing. We'll get into that in just a moment. So as an example, this shows a slide of some work that was done to look at the different forms of sulfur speciation in corals. And you can take a wide variety of different energies through that absorption edge. You can see here are model compounds and the energies that were selected. And most of these energies were selected to try to differentiate contrast between the different sulfate and oxidized species and the different organic and reduced species. We can take these energy maps and do a linear combination fit and get the species maps and see how different forms of sulfur then are distributed in a spatial sense and how that correlates with optical images and optical fluorescence images to see how different structures correspond to different organic compounds. And of course, we can also go and do that traditional spot analysis. Here, there's about 36, 37 or so different spot XANES that were taken. And we can verify that the speciation that we calculate on these maps from the this case, 8 different energies is a valid fit. This is a very powerful technique now to be able to visualize your chemical speciation rather than just looking at these lovely squiggly lines all the time in the graphs. So going back to a geological sense here, what we're trying to now to look at is these real assemblages that have heterogeneities on multiple length scales. We want to take things from these basin scale ideas and rocks that are taken from those into these thin sections on the micron scale and examine these complexities of these samples to better unravel the chemical history of what's happened with sulfur in the geological timescale. And really, this is not really a combination of both these XAS imaging techniques, with standard petrography, and other techniques like SEM, SIMS, and magnetic techniques and bulk scale measurements to get a broad picture of what's happening in these settings. What we're particularly worrying about here in this first, I'm going to show 2 examples, one on more the reduced side and one on more the oxidized side. When we're talking about the reduced side, I'm looking at various pyrites. These all have the origin around the rise of oxygen, so about 2.5 billion years ago. And this is the time when we're looking at basically trying to determine when the rise of oxygen happened. We have constraints on some sides of where oxygen can and can't be, and trying to narrow down these error bars of when this process happened. Now, a lot of what's been done with this is has been done to link the sulfur and oxygen cycles by looking at isotopic measurements. A lot of these work on large hand samples or even core samples, but on a large volume of rock. And we also always assume that these rocks that we're bringing up from depth that are billions of years old really have not gone any post-depositional changes. So by looking at the micron scale texture, this can give us a lot of understanding of what's happened over geological time and trying to unravel some of these complex geological histories. So we'll start with an Archean sample, Archean pyrite. This is one of my favorite samples because it has these 3 pyrites here in this shale, and it kind of looks like this has a SIMS standard in the center here, but it kind of looks like a little smiley face with 2 eyes and mouth and nose. So this is why it's one of my personal favorites. It has the best look to it. These are taken from the northern end of South Africa in the Griqualand West region on a series of cores from the Agaron Foundation. These are coming from this region that's ideally pre-oxygenated, so about 2.5 billion years ago in a distal slope environment. And initially, we did this experiment by just because we wanted to look and see what our new Beamline could do with pyrite. But then we really began to find some very interesting things right away with these samples. So I'm going to draw your attention to this region here, and this compaction rim, the pyrite and the shale here. And by looking at the spectroscopy and putting together an image map, you can see that there's pyrite, which is a very good sign. Noted in blue here, you can see some pyrite, smaller crystallites on the outside here next to this big deposit here, precipitate. And there is these lath-sized little sulfates that go through here. It's a very typical sulfate signature. But what really began to find interesting here was this red spectral signature here, which is found in these locally discrete, say 5 to 25 micron sections along this sulfate kind of inclusion area. And it really had a very odd spectral signature, and for now, I'm labeling it unknown. And this is particularly intriguing now, and it also co-occurs along with these sulfate rims in this particular sample. So what else can we see about this? Well, we combine this. This is now from another sample in a related core. And this comes back to a study that was done where the sample was exposed to nanosims to make isotope measurements, and as well as micro squid to look at the magnetic field. And if we look at the difference between the pyrite nodules, there's some pyrite cements in between. if you look at a sulfur composition map overlaid here, you can see that there's pyrite is full of sulfur, as we might expect. If we take that same region here that is in the paper with the isotopes and the magnetics, we can see that again, it's mostly pyrite, and yet there are these little, again, unknown inclusion spots in those areas as well. And what's very interesting is that these inclusions here match up reasonably well with where we see isotopic anomalies, as well as we see anomalies in the magnetic field of the sample. Pyrite itself is not magnetic, but this particular region here shows an increase in the magnetic field. So what this also then really brings us into is when we're trying to do these multiple techniques between SIMS, magnetics, isotopes, X-ray techniques, we really need to have careful registration between these different techniques to really be able to properly correlate these rather than just the looking by eye to try to make these determinations. What does this tell us, though? So when we look at these types of Archean pyrites, and we look at them in bulk sulfur techniques, you see a wide distribution in the delta 34S and the cap 33S. Typically, if a sulfur isotope measurement falls along this trend line, it shows that it has been exposed to oxygen, so the isotopes, the mass independent fractionation changes. And if you're in a anaerobic oxygen-free environment, you would be in this realm over here. Now, by looking at these rocks in bulk texture, you see a kind of a mixture of both types of distributions, and that can be very confusing to try to say when you had oxygen occurring in the atmosphere. However, if we actually can separate these based on their textures, for instance, so we see intact nodules and things like rims and other portions, that separates out very nicely. So all these late pyrites that show that happened after the rise of oxygen should kind of cluster along this line here, and all the in situ pyrites that were primary fall along this area here. And so the chemistry and the micro and macro scale really do matter. So now what does our mystery sulfide really say about it here now? And this is where we're going to do some calculations here. And when we look at pyrite itself, that pre-edge feature in pyrite is actually a combination of two pre-edges. So it's a 1s to 3D transition here, and a 1s to the sigma star antibonding in the sulfide. Now, to get our mystery spectra, there we go. One possible interpretation of this would be to look at the spectra here and separate this into a series of 4 Gaussians. And this could be described by the sulfur seeing both iron 2 in the disulfide and iron 3. Iron 3 has a lower energy level that brings our lower area down, and the crystal field splitting is higher, so that would create, in a hand waving kind of way, peaks at the lower and upper end. So we wanted to confirm this kind of thought behind this, and we did some preliminary DFT, or time-dependent DFT calculations with Orca, with a very simple 2 or 3 atom cluster of this. And the calculations kind of show us that the peak separations are the right magnitude, and the magnitudes of the 2 pre-edge features are about in the right location as well. So this kind of gives some possible credence to that interpretation, but it'd be really nice to be able to do more detailed and larger clusters to really look at the dynamics of these particular transitions. This is one case where I think these types of calculations really help to explain the spectroscopy that we're seeing, and can really give a nice interpretation to what's happening in this particular system. So I'll leave this portion to here and now move on to the oxidized parts of the systems. So the oxidized end here now is looking at sulfate. Now, in the converse side, looking at sulfur isotopes in sulfates is also very important. That's the other half of the redox cycle. And precipitated sulfates, like in barite deposits, can give us the isotopic signature of seawater throughout time. However, if you try to find the distribution in the geologic record of major evaporite and barite deposits, you have a whole bunch here that occur in more modern marine environments, and you have a large gap in the history of these types of deposits. So it'd be nice if we had a different type of proxy that could kind of fill in these large billions of years of time in between where we have very solid evaporite deposits. Carbonates are a more ubiquitous type of rock that's found in the geological environment, in the marine environment throughout time. And they tend to have small amounts of sulfate that can be incorporated into the carbonate lattice. And this is often called carbonate-associated sulfate, or CAS, as we'll kind of abbreviate it for the rest of this talk. So one real question is, how do we actually fit a tetrahedral sulfate into a planar carbonate space in a calcium carbonate type mineral? Some calculations, dynamic simulations were done earlier by Fernandez-Diaz, and showed that if you try to shove a sulfate into a calcite, it actually has a different structure and a different perturbation than an aragonite lattice. And that the energy of trying to shove sulfate into an aragonite is much higher. You see larger perturbations. The tetrahedron has to tilt a little bit more, and it causes a larger lattice distortion. So one question would be, maybe we could see some of this in our spectroscopy, looking at carbonate-associated sulfate in spectroscopy parts of things. And so this is actually a combined study that looked at both the spectroscopy as well as the isotope shifts that occur by trying to incorporate sulfate into calcium carbonate minerals by co-precipitation. Now, interestingly, there's a small shift here. So calcite is in blue in these, and aragonite is in yellow in these plots here. And there's a very consistent shift to a slightly lower energy of the sulfate peak in the aragonite case compared to the calcite. And there's of course, an intensity relationship that goes with the concentration of sulfate in there, some slight broadening in our peaks, but this constant shift here that's invariant between aragonite and calcite effectively.
2 minutes.
You got it. And there's also then a corresponding isotopic shift that we see where aragonite is very independent across this, where calcite has a isotopic effect. And so the question is, how can we interpret this? And this is one of these really big questions that I have for the DFT community here to actually try to help look at this and explain this particular shift. My first hand-waving type of way is by comparing this to protonation effects on the change of symmetry. This is shown for differences, say, between sulfate and protonated sulfate, where that break in the symmetry breaks the degeneracy of the T2 star orbital in the sulfate and creates a broadening in that peak shift effect. So I'd be really interested to know if this type of effect is similar to how, by shoving this sulfate into a distorted lattice, can we actually model that effect of this energy shift and show that it is or is not consistent with that. But when we look at real samples, of course, it's actually interesting because we can see this in real geological samples. We can see the change between, say, a typical inorganic sulfate, say more calcite forms in these rims in the green bits here around different clasts that show up in secondary calcite textures compared to a more primary, maybe form, distorted sulfate in the center and cores of some of these clasts. So we can actually map out this small shift and see it located in real samples as well. So the next step is to really get a really good interpretation on this, and I think this is where some good calculations come into play. So I'll finish up just by kind of summarizing all that. There's a lot of really interesting heterogeneities in these different environments, both reduced and oxidized sulfates and sulfides. And I think these systems could really benefit from further understanding using computational methods. And can this really give us more insight into the processes of formation and help us interpret other measurements that we do both on the bulk and the micro scale. And I think this actually is really important to understand so we can really make these extrapolations onto larger length scales. And I'll finish up in my last 5 seconds here with thanking the team of scientists that I work with in the imaging program at SSRL, Sharon, Nick, and Jocelyn. And the work I have here shown here is from a set of collaborators from Caltech, WashU, Woods Hole, and the Smithsonian Institute. And with that, I'll take any questions.